make sense? Nonsense. I might talk nonstop, but no going bad-mouthing me. All puns aside, if there's a problem with a messenger RNA and a ribosome tries to make a defective protein from it and gets stalled, this is going to be a big problem. And so your cells have various surveillance pathways that are able to detect problematic messenger RNAs based on ribosomes having trouble trying to make proteins based on their instructions. There are several different reasons why an mRNA might have issues with it, and why a ribosome might therefore get stuck on it. And so there are three main pathways that are able to kind of detect these different kinds of messed up messenger RNAs. And these include the non-stop-mediated decay, where there's no stop codon, so the ribosome doesn't know where to stop making a protein, and so it kind of just plows through where it should stop, and ends up at the end of the messenger RNA and it can't get off. So that's non-stop-mediated decay, and it has various proteins that help it out. Then there is a kind of opposite problem, where nonsense-mediated decay, where the ribosome tries to stop too soon. There's a premature termination codon. So it tries to stop before the end of the protein, and now it's too far away from all that machinery that's needed to actually get the ribosome off, and so the ribosome stalls this way. And then finally, there's no-go decay, where the ribosome stalls for some other reason, some other problem, say, with the messenger RNA transcript. And so that's no-go decay. And so these different pathways, they have different key players, but the bottom line is they all work pretty similarly. They do what you would want them to do: degrade that messenger RNA so other ribosomes don't try to make protein from it and then get stuck as well. So you can cut the mRNA to expose it for degradation. That's going to take care of the mRNA. You want to take care of that defective protein, so you ubiquitinate it. You give it a tag that's going to give it its free ticket to the proteasome, the protein shredder, because whatever the messed up protein that was coming out of that ribosome, we don't want it going around and messing with things in the cell, so we degrade that. What about the tRNA? It might be okay. So basically, there are ways that you can kind of mark the tRNA, give it a little check if all's okay with it, then send it back into circulation. And the ribosome, well, it's probably not the ribosome's fault. So the ribosome, you just need to kind of recycle it. And so there are various proteins involved in these different aspects, but they all have a similar end result, where basically you get rid of the defective mRNA, you free up the ribosome to make more proteins, and then you're preventing more ribosomes from having issues with this mRNA, more making of defective proteins, all those various things. So let's go into more detail. Let me start by saying that this is a very active area of research, and so I'm going to present things as I understand them best to date. I'm also going to point to some recent review articles as well as research papers that help kind of flesh out some of the details. The details are also going to differ in the names and things when research is done in different species, but I'm going to kind of give an overview of how the process works in general. And if you want more of those details, then I recommend you check out the recent papers, and also keep your eye on this area because it's a very active area of research, because some diseases are caused by or involve problems with faulty messenger RNAs or faulty quality control mechanisms. So let's talk about how we can get faulty mRNAs. First off, we just need to do a really quick review of some of the terminology and the overall process of making a messenger RNA. So basically, the original recipe for making a protein is written in the form of DNA in the form of a gene. And so a gene is just a stretch of DNA in these long coiled up pieces of DNA called chromosomes. A region of that gene is then copied into a pre-mRNA, a pre-messenger RNA, in a copy called transcription. This is kind of a letter by letter copying. And so the pre-mRNA is going to include regulatory regions of the gene that need to be removed. And these are removed in a process called splicing. This process called splicing removes the introns, these regulatory regions, from the pre-mRNA and splices together the exons, or the parts of the actual protein-making instructions. This pre-mRNA also gets a 5' cap and a polyadenosine tail, and this then makes it ready to go and bind to the ribosomes. And then the ribosomes are going to travel along that messenger RNA, and they're going to piece together the amino acids or the protein letters
that are coded for by that transcript. So the mRNA is called a transcript because it was transcribed. And it's coding for these various proteins, basically in the form of these codons, these 3 letters. Every 3 letters spells one amino acid, and the ribosome is going to travel along with the help of tRNAs. It's going to piece together those amino acids. And so this is how you end up getting a protein. And so what we're talking about today is when there's problems with that messenger RNA that are going to cause problems with this process of translation. And if they cause problems with this process of translation, well, then you're going to end up with issues where the ribosome is going to get stuck on these transcripts. It's going to be making faulty proteins. Those faulty proteins can go off and do weird things in the cell if they're not removed. If they are removed, that's great, but you don't have to keep removing them, because if you keep making all these faulty proteins, you're going to be hogging up the protein removal machinery, the proteasome. This basically how your cells are able to degrade faulty proteins is through a proteasome system. And if you hog that proteasome system, then you can't degrade other proteins that need to be degraded. Plus, it's really a waste of time and energy to keep making all this faulty stuff and then degrading it. And you're kind of also hogging the ribosomes when they could be making things that can actually work. And so these processes are really important for removing that messenger RNA. And so you don't keep making these problems. So how do you get that faulty mRNA in the first place? Let's go back and think about how we actually made the mRNA. The 2 places where problems can arise are with the transcription and with the splicing. This is assuming that the DNA version is okay, that is. So if there's a mutation in the gene that'll cause one of those problems that triggers this decay pathways, then every time you make a copy of that messenger RNA, you're going to have issues. And this is the source of some diseases where you get various things like premature termination codons every time you make that mRNA. But often what happens is you just have kind of one-off problems in all of us. So it's not like every time, it's not like a disease causing, it's not like every time we make this, we can't make a functional mRNA. It's more like sometimes our cells make mistakes. And so when we're doing the transcription, maybe we introduce a typo, and maybe that typo is going to introduce a stop codon. And so when we talked about those different codons, you had each of those 3-letter words kind of spelling a different amino acid, but some of them spell stop. And so the stop codons is UAA, UAG, or UGA. These are going to tell the ribosome where to stop. And you can have like a point mutation, which is either going to change a stop codon to a not a stop codon. And in that case, then you could end up with something that's like a non-stop decay, or you can have something, you can have a mutation that changes a letter into a stop codon, which we call a nonsense mutation. And this is why we call it nonsense-mediated decay. So this can happen with a point mutation, or it can happen if you have an insertion or deletion where you get a kind of like frame shift. So because you're in these chunks of 3, if you add or delete something that's not a multiple of 3, so like a 1 or a 2, a multiple of 1 or 2, then you're going to introduce a frame shift. Basically, the ribosome, the genetic code is not overlapping, so where it needs to know where to start with that start codon, and then it reads 3 by 3 by 3 by 3 by 3. So where it starts is going to matter. And so if you introduce a mutation that is not a multiple of 3, well, now you're going to get out of frame, and this can introduce a stop. This often introduces a premature stop codon. You can also have problems, so that would be like point mutations or insertions and deletions. Also, if you have alternative splicing, inaccurate splicing. So one of the things about splicing, that's where we remove those regulatory introns, was that you could do it in different ways to get different versions of a protein from the same gene. And this is really important for generating a bunch of diverse proteins without having to take up a bunch of space in our DNA. We have so much DNA as it is. We want to make the most efficient use of what we have. And so alternative splicing allows us to do things like make versions that have a nuclear localization signal in some cells so that the protein goes to the nucleus. And then sometimes in other cells, maybe we don't want it to go to the nucleus, so we just make a cytosolic version. We can do this by making different like alternative splice isoforms and things like this. But sometimes the splicing can actually go wrong. And so you can end up with splicing mutations. This can be caused, again, either by a permanent error in the gene or by kind of a mistake with just the processing or with the transcription. And so what you can end up with is you can get things like including an intron or skipping part of an exon, and these things can introduce frame shift mutations, which are going to cause those problems as well. So various ways in which you can generate mutations that are going to, or generate these problems with the messenger RNA that are going to trigger these quality control pathways. So now that we know how these faulty mRNAs can form, let's talk about how we go about taking care of them. So as I mentioned, there are several different forms of translation-coupled mRNA decay. The terminology can get kind of confusing, and so often like no-go decay or NGD or non-stop decay or NSD, these are kind of more similar to each other than like nonsense-mediated decay. So nonsense-mediated decay is a little different. No-go decay and non-stop decay, these are often referred to as involving like the ribosome quality control pathway or RQC. Although sometimes you see like NMD lumped into, and I'm not exactly sure when you, who calls what. But we're going to go over kind of the basic details of all of these. And so let's start with the nonsense-mediated decay, or this NMD. So basically, nonsense-mediated decay, remember when I said that the mutations that changed one of the codons that actually had the instructions for putting in a protein letter? A nonsense mutation changes that into a stop codon. So one of those codons that tells the ribosome to stop. Now, what happens when the ribosome actually reaches a stop codon is in the process of termination. Basically, these termination factors are going to come in and they're going to kind of pretend to be, one of these tRNAs, but they're going to come in and they're actually going to cut the growing chain off of the tRNA in the ribosome, and therefore release this nascent chain. So the ribosome is moving along, it's the tRNA is bringing it these amino acids, the ribosome is piecing them together. The ribosome gets to the end, a release factor comes and kind of mimics a tRNA, it sneaks in here and it cuts off this chain. The chain gets released, and then the ribosome can do it again. But what happens is that if you have a premature termination codon, you're going to get a stop codon before you should actually have a stop codon. And this means that elongation factor, that release factor is going to be recruited before the end of a protein, before the end of the messenger RNA. And this is the issue. Not only would you make a partial protein, but you actually have problems with the termination, because the termination is actually going to be helped out by things that are happening in the tail. And so there are proteins that are going to be binding to this tail and things like this to help promote the efficient termination. And what happens is if you have a premature stop codon, you're going to be too far away from that tail to actually get that help out. In addition, often what happens is you're going to have an exon junction complex that's left on there. And this is going to help inhibit the process, the termination, and it's going to help lead to assist this nonsense-mediated decay. So what are these EJCs? These exon junction complexes are basically these protein complexes that get left behind during the process of splicing. So when those introns are removed by this complex called the spliceosome, they leave a mark. They leave behind this exon junction complex, which is about like 20 nucleotides or so upstream of the exon junction. So where they splice together those exons, they leave this complex behind. Now, what happens in the pioneering round of translation, which is basically the first round of translation? So the ribosomes are going to use this mRNA over and over and over, and lots of ribosomes are going to use them. But the first time it goes through, we call this the pioneering round of translation. And what the ribosome does is it's going to be translating, but it's also going to be plowing through those exon junction complexes. And so it's going to plow through them all, push them off, and then reach the stop codon. But what happens if it reaches a stop codon before the real stop? And what if it reaches a stop codon before the last exon? There's going to be an exon junction complex there. And if there's an exon junction complex there, well, now it's going to kind of interfere with the ribosome trying to talk to this machinery that's going to be helping terminate it. So when those release factors come in, they're going to actually be interacting with these proteins in the exon junction complex. And the exon junction complex is going to kind of then help promote this nonsense-mediated decay pathway. And so what's going to happen is you're going to get things like ubiquitin ligases tagging that partial protein for proteasomal shredding. So basically it's going to get tagged with ubiquitin, which is this little protein we can stick on the ends of things we want degraded and get them sent to the proteasome, which is the protein shredder, to chew it up. We're also going to use nucleases to chew up the mRNA. So there are actually, we talked yesterday about how basically if the mRNA has this cap in this tail, and if you remove that, then there are exonucleases, which can basically chew up the ends of those RNAs. And basically what can happen then is you get this degradation. In the case of nonsense-mediated decay, you can actually get degradation in different ways, and there are a lot more details that you can see in this paper. This review paper that came out in 2015 is really nice. Nonsense-mediated mRNA decay and intricate machinery that shapes transcriptomes, by Sorek Anderson and Torben Heik Jensen. So highly recommend. So I wanted to show you this paper for a couple of reasons. One is that it helps highlight some of the key players. So you have like the SURF complex and things. Another is that it actually shows you how you can have those multiple decay pathways. Well, one of the really key reasons I wanted to show it was because it also shows how you can get this nonsense-mediated decay, even if you don't have an exon junction complex left there. And this is becoming more and more recognized, is that you can get kind of this nonsense-mediated decay on transcripts even when you don't have that exon junction complex. So initially it was thought that you needed the exon junction complex, and in fact the exon junction complex can play a role. It can assist the process, but it's not necessary. Instead, what's more important, it seems to be, is the distance from the tail. And so the tail is going to be coated with this protein, this poly-A binding protein, which I talked a lot more about in yesterday's post that I'll link to. But basically this poly-A binding protein is going to do things like interact with the release factors. And this is going to help promote the efficient release. If that tail is too far away, then it's going to have a hard from the ribosome when it stops, it's going to have a hard time kind of helping the ribosome actually release that transcript. And so when you're too far away from the tail, then you can have these issues. And so one of the reasons why you could be too far away from the tail is if you have the premature termination codon. But this can also happen just on transcripts that happen to have a really, really long 3' UTR stuff. So there are cases where you can get kind of this nonsense-mediated decay, even if you don't have a premature, say, translation stop codon. But I'm not going to get into that too much. But I do want to mention that although the PTC might not be necessary, there's this protein that's really crucial, this UPF1. And the UPF1 is kind of going to bind non-specifically to the messenger RNA. And the more room you have between the ribosome and that tail, the more chances there are for UPF1 to bind. If there's an exon junction complex, the UPF1 can interact with that and kind of help make all of this come together. But even if there isn't, you can kind of get this non-specific binding of UPF1, and it can kind of then therefore make it so that you can have this process taking place. There are a number of diseases that are known to involve nonsense-mediated decay. So if there's a nonsense-mediated decay can be really, really common because there are a lot of mutations that can cause an introduction of a stop codon. If you get out of frame, it's really simple to kind of introduce a stop codon, which is kind of a good thing. So your cells don't just make a bunch of gibberish. But if that's happening every time you go to make the protein, well, now you're not going to make a functional protein. And even if the protein, maybe you made it almost all the way, but then it stops too soon. Maybe if you can make that partial protein, it could still be useful. Or if you can make a protein with a mutation in it, it could still be useful. But your cells are kind of, the quality control is getting rid of them. And so there are actually different strategies for kind of helping promotes the cells to skip that pathway. So kind of there are translational read-through compounds that allow the cells to kind of ignore the stop codon and just skip over it if there's a mutation. And you might think, okay, well, that's going to be really, really dangerous because it's going to do that in all of the locations. But it turns out that those compounds aren't that efficient typically. And there's often there's actually extra stop codons at the end. So if it goes through a normal stop codon, it's not going to be the end of the world. And so something to keep an eye on is this like PTC read-through compounds, but I'm not going to go into that here. Also, before I move on to the other forms of this surveillance, I also want to mention that although I've been talking about NMD in the context of like diseases, it also plays important regulatory roles outside of disease causing. And so sometimes, for example, it helps regulate the making of splicing repressors and splicing activators. So basically, due to alternative splicing, you can get, depending on how active the splicing machinery is and how much you have, you can get various versions of these transcripts, some of which might have a premature termination codon. And when it has a premature termination codon, then it'll get degraded. And you can kind of control the levels of the splicing activators and repressors and things that you make. So very cool stuff like that. More on it in this article. But now I want to move on to the other forms of ribosome-associated quality control. This like no-go decay, where you have a stalled ribosome for some reason, and non-stop decay, where you have a missing stop codon. So before we're talking about having like too soon of a stop codon, and here we're talking about not having a stop codon, which again can be caused by if you have a mutation in that stop codon, if you have a problem with your splicing, if you have premature adenylation or something like this. And this is going to lead the ribosome to not stop and to kind of plow through into the end. Often these are going to be associated, like the signal for these is going to be like not just one stalled ribosome, but actually stalled ribosomes causing collisions. So basically, although we often I'm drawing these ribosomes as being single ribosomes traveling along an mRNA, what happens more frequently is that you actually have polyribosomes. So you have polysomes, where basically what you have is you have multiple ribosomes traveling along that mRNA at the same time. This is going to be much more efficient. And it's also going to give us a way to sense a stalled ribosome, because what happens is if one ribosome stalls, it can kind of get the other, it can lead to a collision when the ribosome behind it comes along and can't go farther. Okay, but wait, why am I drawing like chapels right now? I need to switch to another metaphor I like to use. So basically, we've been used to showing the ribosome as something like this, where you have this large ribosomal subunit, that big thing on top. You have this small ribosomal subunit. You have these three sites where the tRNA can live in. You have the E, P, and A. A new tRNA is going to enter the A site. The growing chain is going to be held with the P site. You're going to get transfer of the chain from the P site to the A site. Then everything's going to shift over with the help of these elongation factors. You get to the end, a release factor comes in and helps release things. So that's what is going to happen in this process of translation, and this is how we typically think about it and draw it and sketch it out. But I also like to think of translation as kind of being a wedding between those amino acids that were added. So if instead of thinking of the ribosome like this, you think of it like this, you think of it as a chapel, you can basically have this large subunit and this small subunit, and the mRNA is going to be like your road. The tRNAs are going to be kind of like limos, which are transporting these amino acids. And which amino acid there is going to be specified by kind of like a parking spot, which is going to be the codon. And when you make that growing chain, it's going to be kind of coming out of the chimney. So the ribosomal, the nascent peptide exit tunnel, the NPET, is going to be analogous to your chimney. So in the case of this ribosome, we have the peptide coming out of the chimney. We have these ribosomes stalling due to some like problem in the road. So some problem with the mRNA, say. And this can trigger no-go decay. The importance of this stalled, like these collisions and things, is that it's going a variety of reasons, but it's going can be sensed, and this can lead to the ubiquitination of the ribosome. So not of the peptide, we'll talk about the peptide later, but the ribosome can actually get ubiquitinated, which doesn't lead to its destruction, but can lead to it getting kind of split into two. And this splitting is going to be really important because it's going to be exposing interaction sites so that this ubiquitinase, this ubiquitin ligase can come in and it can ubiquitinate the chain, and then that chain can get destroyed. So this ribosome collision thing is a really active area of research, and I highly recommend that if you want to know more, you check out Rachel Green's research. She's at Johns Hopkins. Fun fact, she was on my dissertation committee, my thesis committee in my grad school. And she's really, really cool. And she's done some really awesome research studying how kind of stalled ribosomes and collided ribosomes are detected. So she also has some cool videos on YouTube. So I recommend you check out her work. But anyway, so we have those stalled ribosomes, and then the cells are going to sense that. They're going to split apart that ribosome. So they're going to split apart our chapel. But we have that peptide that's going to be stuck in the large subunit. So it's going to be stuck in that tunnel. And so what we need to do now is we need to actually target that nascent peptide, that growing peptide that's in the tunnel. We need to target it for degradation, which we're going to do through ubiquitination. And then we have to deal with this mRNA. So we have to kind of like destroy this road. And then we also have to figure out what we want to do with that tRNA. So with like our limo in this analogy. And so basically, here's another review article, and you can see that there's going to be a lot of different players involved. And one of the key ones is going to be this Listerin, or this LTN1. So Listerin, not Listerine, but this is going to basically be the ubiquitin ligase, or that ubiquitin adder that's going to interact and ubiquitinate the growing chain. So you can see this nice review article. It looks kind of like this. See, it's kind of like it looks kind of like a coffee cup handle. But what it's going to be doing is, see, this is why you needed it to have removed the bottom the small subunit, because it would get in the way of the binding site for this Listerin. But it's going to bind. It's going to then ubiquitinate this growing chain. And this is how you can get this chain, take it straight to the proteasome and degrade it. But what about that tRNA? So that tRNA might still be okay. But there could also be a problem with the tRNA. Problems with tRNA can cause the ribosome to stall. So we want to make sure that that tRNA is okay before we stick it back into circulation. And we also need to kind of release the tRNA from the growing chain. And so the proteasome is going to take care of the protein part, but this chain is actually still attached to that tRNA. So we need to cut them apart. So remember that in the context of having this stalling, you're not having that peptide chain release like it would be if you had an elongation factor come in. And since you don't have an elongation factor come in and cut off the chain, what's going to happen is you're going to have that still attached, and so you need to cut it off. When the release factors cut a peptide off from a tRNA, normally it's going to be done by cutting the peptide tRNA linkage. But what happens when you have this is you're actually going to cut the tRNA itself. So you're going to kind of cut the part of the tRNA that links up to the peptide, but in the tRNA stem. And you're going to cut it in a way that leaves a weird mark, which is like this cyclic phosphate. And so in order to actually be used, this tRNA is going to have to be converted into a form that can like function, and it's going to have its adapter put back on. So basically you're cutting the adapter that allows it to link to the amino acid, to the peptide chain. And so you need to add that back on. It's like this CCA thing. So basically the sequence that's on the end of all these tRNAs it's going to get, that allows it to serve as an adapter to link up to these peptides. So you can have the same like adapter adder add on to and fix all these tRNAs if they're okay. And if they're okay, then they can go back into circulation. So you get this like recycling of the tRNA thanks to this tRNT1. So here's a cool paper from a few years ago I got to hear about at a meeting I went to. So I recommend you check it out if you want to learn more about how those tRNAs are going to be recycled. But going back to the idea of ubiquitinating that growing peptide, so basically, if you have, in order to get ubiquitinated, you have to have a site that can be ubiquitinated. And typically ubiquitination is going to happen on lysine residues. So lysine is one of the amino acids. Sorry for using, when I said the word residue, basically that's just one of the amino acids. Once they link together, you call kind of like the remainder or residue. So when it's free floating, we call it an amino acid. When they're linked up, we call these individual things that once were amino acids residues, although we often use the term amino acid still interchangeably. It's not technically still an amino acid because it doesn't have the X acid group and things like this, but that's just a technical note, and I just wanted to clarify in case you weren't familiar with the term residue. That's what we mean. That used to always trip me up. But anyway, so you need to have an accessible lysine to have this be added onto. There are also some other places where you can get ubiquitin added, but it's typically on a lysine. And so you need to have an exposed lysine that's going to be accessible when the listerin is bound. It needs to have a lysine that it can then access. So if there's a lysine already there, great. But if there's not, what are you going to do? You need to make it so that there's a lysine there. One of the ways that you can make it so that there's a lysine there is by cattailing. So basically, you add untemplated amino acids, these carboxy terminal alanine and threonine. There's this complex, this cattailing complex, that what it can actually do, this like RQF2, it can actually come along and it can add a bunch of adenines and threonines, and this is going to push the tail out. And so this is going to then expose any lysines that were kind of stuck in the tunnel. Now they can be pushed out, where they can then get ubiquitinated. What about in the case of nonstop decay? Well, here you're kind of one step ahead. If you're missing a stop codon, well, now what you're going to be doing is you're going to be going through that poly A tail. And that poly A, it's, if you go back to our codon chart, if you have AAA, well, that's going to be a codon that's that codes for lysine. So right now you're going to be putting in a whole string of lysines, and eventually you're going to be pushing those lysines out of the tunnel. And when you push those lysines out of the tunnel, well, then they're going to be exposed. They can get exposed. They can get then get ubiquitinated, and this is going to then lead to the degradation. And so in these pictures, I've shown the ubiquitin when the ribosome is still like in its full form, but really this is going to be happening once you have the ribosome actually be dissociated. Then you're going to get this ubiquitination because you've had the sites exposed that can then be ubiquitinated. You're going to get that tRNA cut off and all that good stuff, but the ribosome is going to be dissociated first. Okay, so that is going to be the basics of this translation coupled RNA decay. So you have nonsense-mediated decay, where you have a premature termination codon. The ribosome is going to be trying to so the ribosome is going to meet a stop codon. That's going to trigger the recruitment of a release factor. Normally, the release factor is going to be communicating with proteins like things that are binding to the poly A tail. But in this case, those release factors are going to be either communicating directly with this exon junction complex, or they're not going to be communicating with that tail because it's too far away. And instead, they're going to be communicating with other proteins that can help kind of serve as surveillance by binding to this sequence. And then if they're if there's a termination, premature termination, those complexes like this UPF1 can then, or these proteins like this UPF1 can then interact with the release factors, cause the recruitment of these complexes that can then cause this whole process to take place, where you then degrade the mRNA and recycle the components. So that's nonsense-mediated decay, and it's caused by premature termination codons. You can have the opposite problem where you don't have a stop codon. In this case, the ribosome is going to plow through, reach the end, and not be able to get released. Or you can have a problem where a ribosome is kind of just stalled for some other reason. In either of these cases, here what you're going to have to do is you're actually going to have to detect that stalled, that ribosome that's stuck, which is often done through sensing collided ribosomes. You then separate the ribosomes, ubiquitinate the messed up partial protein or overmade protein, send it to the proteasome, recycle the ribosome, check the tRNA to see if it's okay, and if it's okay, then you can reuse it. And so that's nonsense-mediated decay, NMD, Go Decay, NGD, and NonStop Decay, NSD. And those are the three main kind of translation-coupled mRNA decay pathways. Sometimes these are referred to as these surveillance pathways, and they help make it so that your cells doesn't keep trying to make faulty proteins from faulty mRNAs and jam up all the machinery and cause problems. And so hope that helped you understand, and I'll post links to those papers if you want to know more.